Welcome to the video tutorial series on genomic tumor testing patient reports, brought to you by the Genomic Education team at the Jackson Laboratory. This short video will walk through the genomic marker interpretations section of the patient report. Back to the patient report for our melanoma patient Tony. In the genomic marker interpretation section of the report, each clinically significant gene variant is described in more detail. At the top of each section, there is the gene name, the variant name, the VAF copy or the variant allele frequency, the read depth, the functional impact of the variant, as well as the type and class of the clinical significance of the variant. Following this information, there are a few paragraphs getting into the detail of the effect of the variant, its connection to cancer, a description of the protein each gene encodes, and its function in the cell normally, as well as a brief summary of its connection to FDA-approved therapies or experimental therapies. Let's look at the first gene variant listed on our patient report. For Tony, we see the BRAF gene and its name PV600E. The P included in front of the variant name indicates that the variant is impacting the protein that this gene codes for. In this case, the variant is an amino acid substitution. The 600th amino acid in the BRAF gene, which is normally the amino acid valine or V, is now a glutamic acid or E in the variant. Now, if the variant had resulted in a stop codon, this would be indicated by an asterisk. A frame shift would have been labeled with the letters FS. An insertion, that is a duplication, would have had the letters DUP in the variant name, and a deletion would have had the letters DEL in the variant name. We are only going to investigate variants that impact coded protein. Let's move on to VAF copy, which stands for variant allele frequency. This is an indication of the number of copies the variant appears in the tumor genome. Next-generation sequencing platforms provide an output of sequence reads, which refer to the specific sequences that match a location in the genome. Sequencing instruments calculate the number of reads detected for each specific genomic location. Looking here at the BRAF gene, we see that the VAF copy is listed as 8%. This means 8% of the sequencing reads for this genomic location contain the variant copy. And this indicates that only a small fraction of Tony's biopsy tumor cells contain the variant. Had the VAF been 50% or greater, this could indicate that all of the tumor cells have one variant copy and could potentially suggest that the variant is present in the patient's germline. This would need to be confirmed using germline genetic testing. Remember, a person is a diploid organism and has 2 copies of every gene in their somatic cells. So a VAF of 50% indicates that 1 copy of the gene in each cell is a variant copy. The depth indicates the sequencing read depth. The greater the read depth, the greater the genomic coverage at a specific region of the genome that is being sequenced. More coverage equals more confidence in the read calls detecting any variants. The read depth listed on Tony's report, 112x here for BRAF, and 912x for P10, not shown here, are both very high read depths, providing confidence in the presence of these 2 variants. The functional impact describes how the variant is affecting the protein the gene encodes. For protein changes, there are 3 possible functional impacts: gain of function, loss of function, and neutral. Typically, with clinically significant gene variants, the impact will either be gain or loss of function. This is confirmed using cellular and biochemical experiments, but To describe it simply, with respect to gene variants that drive the initiation or progression of cancer, this classification depends on the role the protein normally plays in the cell. For instance, if the protein is responsible for stimulating growth or signaling for proliferation or cell division, then a pathogenic variant will typically be gain of function, then leading to greater ability to stimulate growth and division. In the case of a tumor suppressor or a gene that slows growth or is involved in DNA repair, a loss of function will lead to less protection in the cell, creating more opportunity for DNA damage, contributing to loss of genomic stability, which can also contribute to cancer progression. Every now and then, a variant will appear on a report, but the protein change is not understood or is deemed neutral, meaning that it does not yield a gain or loss of function. The clinically significant class and type will be discussed in another video in this tutorial series. Lastly, the paragraphs below describe the gene and the gene variant with cited sources from the scientific literature as indicated by the PMID numbers for the sources used. For each gene, the coded protein information is detailed, along with the normal function this protein plays in the cell. Then there is a description of the variant and how it impacts the protein. There's also a description of the types of cancer this variant is observed in frequently. For instance, for BRAF, we can see that 80% of malignant melanomas have a V600E BRAF variant. This variant is also commonly detected in thyroid, lung, prostate, and colorectal cancers. There's a quick description of the therapies used to treat individuals with the BRAF 600E variant, but this will be covered in more detail in the therapies video in this tutorial series. Thank you for listening. For more information on genomic tumor testing patient reports, continue watching the other videos in this series. Also, check out the other resources provided by Jack's Genomic Education on our website.